So let's finish up the digestive system. We're going to look at the gallbladder and some other things that are left. All right, so the gallbladder is this pear-shaped organ here. It is on the inferior surface of the liver. It's connected to the cystic duct and joins the hepatic duct, which are both, here's the hepatic there, cystic there. See, it joins those. The function is to store bile in between meals and then concentrate it by reabsorbing water. It also you can have issues with it when you get things like gallstones, right? We've heard of gallstones. We know they're very painful. These can form whenever bile salts, pigments, and cholesterol are in a very high concentration so that they actually precipitate out. And this can occur along with gallbladder inflammation, which we call cholecystitis. So what exactly is bile? Bile contains bile salt, pigments, electrolytes, cholesterol. Bile salts serve to emulsify fats. They break down fats. They surround them, and we'll look at emulsification here in a moment, and help us so that we can break them down. They also help to absorb fatty acids, fat-soluble vitamins, and cholesterol. We have bile pigments, and this is bilirubin and biliverdin. We break bilirubin down in order to make it smaller so we can excrete it as biliverdin. When you're not able to do that, it creates problems. And we call that jaundice. Bilirubin is a byproduct of hemoglobin from red blood cells. And so infants having jaundice is something that is a problem just because the infants haven't fully formed some particular mechanisms. But when adults have this, it's a much greater problem. When chyme that is high in fat content enters the duodenum, cholecytokinin is going to stimulate the gallbladder to contract, and then it's going to release bile. So bile salts help digestive enzymes, and they help the absorption of fatty acids. They do this by forming what we call micelles, not like missiles, but M-I-C-E-L-L-E-S. And this is where water surrounds and makes the fatty acids soluble in chyme and easier for epithelial cells to absorb. So basically they're reducing surface tension, and they are taking this fat globule here, and the bile salt is going to surround it, and you can see all the way around that process of emulsification and break them apart into small droplets. And this is basically what happens whenever you put soap in the sink with greasy water. It breaks it all apart, right? That's emulsification. Okay, so without the aid of bile, you will very poorly be able to absorb lipids. It just won't work very well, and that will cause vitamin deficiencies because lipids help us to absorb fat-soluble vitamins like A, D, E, and K, and then we can actually recycle and use those bile salts again and return them to the liver. From the stomach, food is going to pass into the duodenum, then go through the jejunum, and then the ileum. Let's trace this through. What's a good color here? All right, so food's going to come from the stomach on through the duodenum, or duodenum if you would like to call it. It's going to go through here to the jejunum, all right, and then it's going to finally come down here and reach and pass through the ileum. Going to receive the secretions from the pancreas and the liver here, and that's going to complete the digestion of nutrients. So digestion is completed here, all right? It's going to complete digestion of those nutrients, and remember now, once it is liquefied, it is called chyme, and then transport the rest of those onto the large intestine so we can get rid of the waste products. We also here have a good picture of some mesentery, all right, and the greater omentum here that drapes kind of over the large intestines. Here's a really good illustration of the greater omentum here, and the greater omentum has a function. It has a purpose. The purpose of the greater omentum is to prevent parietal and visceral peritoneum in the abdominal cavity from sticking to each other, sticking to the abdominal wall or to the ileum. So let's go ahead and look at the structure of the small intestines. What is the wall of the small intestines like on the inside? It's got tiny projections that we call villi, and these are mucosal. We do have mucus lining them, and these serve to increase the surface area. So if we have a surface that is like this, or a surface that is like this, which one will have more surface area? Well, if you picked this one, you're absolutely right. So what does surface area do for us? Well, if we increase surface area, we increase the amount of area that we're able to absorb nutrients, okay? We can absorb nutrients in this surface area with more surface area. And so each little villus is singular, villi is plural. So each individual villus has simple columnar epithelium. You can see here you have some blood vessels. You can see there is a lacteal or a lymph, it's the green here, a lymph capillary. We have some nerve fibers that innervate here as well, and there are some goblet cells. Remember what a goblet cells do? They secrete mucus, right? Mucus is important because it's protective. And then these columbar cells, the villi actually have microvilli, and we'll see that in the next slide, so you don't have to wait for me to try to draw that out for you. Okay, so we'll look at the microvilli. And these, we've got the intestinal glands here. They're also called crypts of Lieberkühn, and these are located between the bases of the villi. And then we have what we call plicae circulares, and these are circular folds of mucosa, and we're going to look at those on the next slide. They're intestinal folds, okay? Villi invite microvilli, then there will increase the surface area. Can't talk today. So here you can see the plicae circulares, the villi within there, and how that forms the microvilli. And so each villi also on it has projections, microvilli. So that really increases our surface area. So what type of secretions does the small intestine have? Well, you probably haven't ever thought about the small intestine actually secreting. We think about that as just being absorption, which is of course a very important role, but in order for absorption to occur, we have to have some things like enzymes to help break things down. And we also need that protective mucus, that layer of mucus, all right? And so we have a lot of mucus secreting goblet cells. Then we also have some that are specialized mucus secreting cells. These are glands. We call them Brunner's glands, and they secrete a little bit thicker, more alkaline mucus when certain stimuli occur, all right? We've got digestive enzymes that are embedded right in the membrane, right where we need them, to break food down before absorbing it. So we have things like peptidase, and remember ASE is how we recognize that we have an enzyme. So peptidase, peptid means peptide, so peptidases are enzymes that help to split or break down peptides into amino acids. All right, so remember, proteins have to have several things to break them down because they're so large. We also have sucrase, maltase, lactase. Well, those should sound kind of familiar. ASC means it's an enzyme. Well, what are those enzymes working on? Well, sucrase helps to split disaccharides like sucrose. Maltase splits a disaccharide into the monosaccharide maltose, and lactase will break down lactose, okay? And so we also have intestinal lipase, and that's going to split fatty acids and glycerol, okay? So it's going to break those down. All right, so those are the enzymes here. Those are really important. Be sure that you know these enzymes right here. And then here are the hormones that are secreted by the small intestine. And we did talk about these in previous slides, but they're worth repeating. We've got enterokinase, and that is going to take the inactive form trypsinogen, remember ogen means precursor molecule, and convert it to trypsin. And then the trypsin will go on and trigger a few other things. And then we had somatostatin that's going to inhibit hydrochloric acid secretion by the stomach. Cholecytokinin, remember that is CCK, cholecytokinin, and that inhibits gastric glands. It will stimulate the pancreas to release some of the pancreatic juice and to stimulate the gallbladder to release that bile, which is important. And then lastly here we have secretin, which stimulates the pancreas to secrete those bicarbonate ions. So remember that those are to help act as a buffer. So we have to regulate these secretions, right? Mucus acts to protect the intestinal wall. We talked about it a little bit, but I didn't quite point that out. It protects the stomach, we've already said, from acids, and it increases the responses to mechanical stimulation when you have an irritant like high gastric juice. And so that is stimulated by the presence of chyme. So when chyme enters the small intestine, we then are stimulated to secrete that mucus. When you have distension in those cells of the wall, that will activate the nerve plexus, okay? And so that sends signals to the parasympathetic reflexes that will also help to trigger the release of intestinal enzymes. So actually, as it begins to fill up. All right, so goblet cells secrete when chyme enters, and then as it fills up, distension in the intestinal walls will activate some nerve plexuses, which will send parasympathetic reflexes and will trigger the release of more intestinal enzymes. So we've already said villi increase surface area. That is so important. Villi and microvilli increase surface area. Why is my pen not waking up? Increased surface area, okay? They make the small intestines really the most important absorption organ of the alimentary canal. This is where the most absorption occurs. Very, very effective, and there's very, very little material that we don't get absorbed at the end because we have so much surface area that will absorb these things, okay? So, monosaccharides, which are simple sugars, and amino acids, how are they absorbed? These guys are absorbed, and you need to know where they're absorbed to. So this part right here, this is important. Simple sugars and amino acids is going to be facilitated diffusion and active transport into the blood, all right, directly into the blood, which is a good thing. That's how we get that quick energy, right? Fatty acids and glycerol, remember, need several steps, all right, because they're large. That's going to go into the lymph and the blood, all right? And then electrolytes are going to move in several ways: diffusion, osmosis, and active transport, and those are going to go straight into the blood. This is a really good chart from your textbook. You need to take this and make some flashcards of it, all right? And so mostly I just want you to focus on how it's absorbed and where, which part of the alimentary canal, and then where is it going to, okay? Is it going to the blood or the lymph? All right, so let's look at this picture here in the top. We're looking at digestion, breaking down a complex carb. There's a disaccharide there, maltose. So here we have maltose. It's breaking that complex carb down into simple carbs. So it's splitting it into two glucose molecules so that they're small enough to be absorbed. In the middle here, we're looking at protein digestion. That begins in the stomach via pepsin, okay? Peptide is triggered by pepsin, and it is completed in the small intestines. So remember, peptides take multiple steps. Start in the stomach, end in the small intestines. Larger molecules are broken down into amino acids, all right? And then here at the bottom, we're looking at fat digestion, okay? Fat digestion is done almost completely in the intestines, Here we're looking at how fatty acid molecules are broken down. So we're going to start over here in step 1, all right? So fatty acid molecules, these are skin cells here, all right? So these are epithelial cells, I should say, not skin cells, but epithelial cells. So each little block there is an epithelial cell. You can see on this side we have some villi here, all right? So the fatty acid molecules are going to dissolve in the epithelial cell membranes of the villi. So they're going to go through the villi. You can see that in this gray picture here, through the villi, and will diffuse through them. Okay, you see how they're diffusing through here, all right? The endoplasmic reticulum of these cells, of the epithelial cells, use the fatty acids to resynthesize fat molecules similar to the ones that were digested. Okay, so they kind of mix them up, put them back together. So the fat then collects in clusters, and that becomes surrounded by protein. The larger molecule now is a lipoprotein, so lipid and protein, fat and protein, and we call that a chylomicron. Okay, that is here. Chylomicron, and this is important. This will be on your test, all right? And the chylomicron then will make their way to the lacteal, which remember is part of the lymphatic system of the villi, and then we will have contractions of smooth muscles in the villi that's going to help empty the lacteals into cisterna, and that is an expansion basically of the thoracic duct. So it's going to the thoracic duct, not duct. The lymph carries the chylomicrons into the bloodstream, basically. So we talked a little bit about movements of the small intestine. We talked about the peristalsis, which is the wave-like movement going down, and then we talked about the mixing and turning, which is kind of more mixing everything up, right? So that's segmentation. That's the ring-like contractions, moving the chyme back and forth and back and forth. So if the small intestine gets really, really distended or really, really irritated, you can have, and you have probably experienced this in your life, what's called a peristaltic rush. And that's when you have to run to the bathroom urgently because you have diarrhea. So if it becomes too full or irritated, it's going to flush everything out really quickly. We also have the ileocecal sphincter, which joins the small intestine to the large intestine. And another point about the large intestine is it's named large intestine because it's much larger than the small intestine, but important to point out, I guess. So moving from the small intestine to the large intestine, we already said it's named large intestine because it's larger, right? We have 5 parts here, and it's important that you know these 5 parts, okay? And so the first one is going to be the cecum, okay? So the cecum. We also then have the colon, and we have ascending colon, transverse colon here. So ascending colon, transverse colon, descending colon. And then we also have the S-shaped, kind of S-shaped sigmoid colon. That's where the S comes from. And then next we have the rectum and the anal canal, all right? And then at the anus, that is the opening, and that is surrounded, of course, by sphincters. We have an internal and an external sphincter there. And then we have the variform appendix that is attached to the cecum, and that contains some lymphatic tissue. So it does have a immune system function. So the large intestine does not really have any digestive function. It does absorb some water and some electrolytes. We do have goblet cells over here, so we have some mucus secretion there, and that's protective as well. This is where we have our intestinal flora or our good bacteria. This helps to maintain a balance in our system. It also, of course, forms feces, which is our waste products, and carries out defecation, which is basically just when you poop. Movements of the large intestine are very, very similar to those in the small intestine. It's a little bit slower and a little bit less frequent than the small intestine, but we still have peristalsis and we still have some mixing movements. You will usually have a large mass movement after eating. This is, of course, when you feel the urge to go to the restroom. We have what we call the defecation reflex, and that is going to relax the internal sphincter first and then the external sphincter. All right, so all you wanted to know about poop and more. What are feces made of? Well, they're made of everything that's not digested or absorbed, which is going to be some water, some electrolytes, mucus, bacteria, bile pigments that are altered by bacteria actually give the color to poop. And then, of course, we have undigested material like cellulose that we're not able to digest. It's going to have a pungent odor, of course, you know, sometimes it does, and that's often a side effect or a byproduct, I should say, of bacteria. Some of those products are hydrogen sulfide, which is that yellow, gassy, stinky, gassy. I say yellow, I was thinking of sulfur. Indole, ammonia, and those are some smells that you are kind of familiar with. So what happens to the digestive system as we get older? Your teeth can become sensitive, gums may recede, and of course we often lose teeth. You can have more heartburn, more constipation, nutrient absorption decreases. We've talked about not being able to absorb your B12 as you get older. So, you know, your accessory organs age, but in general, our whole system slows down and just doesn't work quite as well as it used to.